Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, as you know that we have already started a series on developmental biology, and today we have brought yet another lecture in the same series for you. In this lecture we will talk about teratogenesis. To discuss this topic we have with us our subject expert Dr. Varnendra Singh Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Hindu College, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome, Sir.
Thank you. So today I'll be talking about a topic known as teratogenesis. In our daily life or in our neighborhood, we must many a times we come across individuals who appear to be malformed or they are different from the ordinary individuals in their physical appearance or their functioning of the biological system. These individuals, how they are born and what are the underlying causes which result in the birth of such individuals. Today I'll be discussing that in this particular lecture, and the objective for this particular lecture would be to discuss about various mechanisms which lead to production of such individuals and what possible role various kinds of agents or chemicals or physical agents like ionizing radiation play in creation of such individuals. So the learning objectives for this particular lecture would be that after this lecture we should know, we will be able to tell that what effect do teratogens have on development of embryo and how these teratogens lead to formation of aberrant individuals. And the reference for this particular topic I have taken from some of the input I have taken from the book Developmental Biology by Scott F. Gilbert. So starting with historical perspective, what exactly is teratology or how it came into existence, what exactly is this science or this field of study. So the birth of malformed individuals has been known from times immemorial, because through our ancient literature or scripts we come to know that these individuals were born again and again, and these were mentioned as monsters in these historical texts. But were they actually monsters? Were they different from normal humans? This we came to know later on, that behind the formation of such individuals are underlying causes, which could be biological causes, which could be genetic causes, which could be physical causes or chemical causes. So since time immemorial we know that congenitally malformed individuals keep on getting born, and these individuals were earlier regarded as evil or monster, but actually that was not the case. These individuals were not evil. Over a period of time, individuals, scientists started to go deep into this subject and study why these individuals were born, and so was born the science of teratology. Teras basically, or teratos basically means monster, and logy means study of. So basically this word literal meaning is study of monsters, but it's actually not the study of monsters. It is actually the science which basically deals with study of development of congenital malformations in individuals. What exactly is teratogen? As the name suggests, teratogen is any substance which alters the embryonic or fetal development, and it leads to alteration in structure and or function of the organ. So basically any substance which leads to change in the structure of the organs of an organism and the function of the organism, function of the organs of the organism, that can be termed as teratogen. A teratogen can cause both the structural and functional change, or it might cause only structural change or functional change. So how the world came to know about existence of these teratogen? What exactly was the study? Basically there were studies going on, and the final proof which came in this particular respect was the study by Rachel Carson. She published a book known as Silent Spring in the year 1962, and this book basically dealt about the use of various pesticide and insecticide and their effect on the animals. So Rachel Carson came out with this particular view or result that the use of pesticide DDT led to destruction of birds' eggs and prevention of reproduction in many species. So this was one of the study which led the scientific world to focus on the harmful effect of these chemicals. Then another such study that in the same year 1962 there was a scientist Lenz who discovered that a sedative drug known as thalidomide, which was extensively used for managing pregnancies, was causing limb and ear abnormalities in fetus. So in the same year there were two studies which came out with conclusion that certain chemicals were leading to aberrant development or they were leading to formation of aberrant organs. After that, this particular science gained motion, and more scientists started studying effect of various chemicals on the embryo during development. In 1964, a virus known as rubella spread across America, and though the adults showed very mild symptoms, the fetus infected by this particular virus became either blind, deaf or both. That is, if a pregnant woman got infected with this particular virus known as rubella, then the fetus which she was carrying, if it got infected by this particular virus, then that particular individual would be born as a blind and deaf, or blind or deaf, or both. He or she might be suffering from both the conditions. So these individuals also some of them had heart defects and mental retardation also. So this particular event in the year 1964 established the link of the external environmental agents and the developmental malformation. So what was earlier a notion now became established because of this particular study, or this particular study in the field, that environmental agents could lead to malformation in the embryo. So what exactly are malformation? At the time of birth, if an individual suffers from anatomical or morphological abnormalities, it will be termed as congenital malformation, because individual is born with these malformation right from the time of birth. So it is congenital. That means it is born along with the individual. These abnormalities, either they may be visible on the external surface, that is on the body of the organism, they might be directly visible. There might be a malformed nose or a malformed ear or malformed spine, or these abnormalities may not be visible on the external surface, but they might be present in the internal organs, in the structure of the organs or the function of these organs. So these malformations account for about 20% of the deaths in prenatal period. Prenatal period is basically before embryo gets implanted in the uterus. So these malformations account for about 20% of all the deaths which occur in the prenatal period. And about 3% of the newborn infants, they have major malformation at the time of birth. That is, all the individuals which are being born, about 3% of them have major malformation which will lead to their death in the coming time. That means they do not survive to adulthood. And about 3% will be having minor malformation which might get detected later in life. So the percentage of malformation is quite high, and it is a miracle that individuals are born hale and hearty without any malformation. So that requires an interplay of various factors which assures that any individual is born in a perfect shape without any malformation. So there are a number of causes for these particular congenital malformation. There is no single cause, a multiple cause which might be acting at once, or they might be acting in series, or there might be a single cause which can lead to formation of these particular abnormalities. And what are these causes? These are basically chromosomal abnormalities, that is abnormalities in the number or structure of chromosome. These might be gene abnormalities, that is a mutation might occur in a specific gene which causes a malformation in the entire organ system or the tissues of the organism. Then there might be environmental factors which are causing this particular malformation, and these environmental factors could be drugs, this could be insecticides, this could be infectious diseases which are caused by bacteria or viruses, and apart from that it could be a multi-factor cause, which is in fact the case for most of the malformation, that it is caused by multiple factors. So these are some of the causes which lead to congenital malformation. Now, what are different types of abnormalities which result from exposure to these particular agents? These abnormalities can be summed up as malformations, disruptions, deformations, syndromes, and association abnormality. So, what exactly are these abnormalities? I will be talking about now. Malformations. So, malformation basically occurs during organogenesis. Organogenesis is basically formation of organs and organ system during embryonic development, and if there is any aberration in formation of any organ of a specific organ system, that will be termed as malformation. So, this particular type of abnormality occurs during formation of, or during the time when the organ system are getting formed. And this particular abnormality results in complete or partial absence of a structure of a given organ or alteration in the structure of the given organ. So, in a normal individual, an organ will be formed of a definite shape and structure, but whenever malformation occurs, this organ will not be having its normal shape. It might be missing a part, or there might be change in the shape and structure of this particular organ. So, some examples are holoprosencephaly and caudal dysgenesis. These are two of the examples of malformation. Now, what exactly happens in holoprosencephaly? Normally, there are two, the prosencephalon, which is the part of the brain, it gets divided into two hemispheres in a normal individual. But in holoprosencephaly, this particular separation into two hemispheres does not take place, and this particular part of the brain, prosencephalon, that is the forebrain, it remains as a single unit. So, there are, as can be seen in this particular panel, there are different degrees of severity of holoprosencephaly. As can be seen in the left hand side is the normal brain, where there are two frontal lobes, whereas in the extreme form there is alobar, this holoprosencephaly, where there is no separation, there is a single lobe, there is no separation into two hemispheres or two lobes. And these are the individuals which are suffering from this particular type of malformation. Now, another example is caudal dysgenesis, wherein normal gastrulation does not take place in the caudal part, and the individual born is having defect in the regions of the limbs, and mostly these individuals do not survive. So, this is an example of caudal dysgenesis, where the lower part of the organism is not properly developed, and most of the time these individuals cannot survive. Now, second type of congenital abnormality is disruption. What exactly is disruption? It is basically a morphological alteration of already formed structure. So, a structure is already formed, an organ is formed, and there occurs a disruption, or there occurs any alteration in the shape of that particular organ, then it will be termed as disruption. And these disruptions or these malformations occur because of certain disruptive processes, like vascular accident leading to bowel atresia and defects produced by amniotic bands. Amniotic bands are basically bands formed around any organ of this particular layer, amniotic layer, and which leads to restriction of the blood supply to that particular organ. When there is restriction of blood supply because of formation of band across that organ, it leads to the organ might not develop properly. So, if an amniotic band form around the organ, it will not be developing properly further on, or if it is developed properly, then it will stop growing from that point onwards. So, this particular figure shows how, because of formation of amniotic bands around the hand of the individual, it led to malformation of the hand of this young one or this newborn. Now, the third type of malformation is deformation. Deformation occurs because of mechanical forces which mold fetus over a long period of time. Now, mechanical forces lead to change in the structure of the fetus, and most of the time these involve musculoskeletal system. And these deformation, many a time they might be reversible postnatally. After the birth, the organ might attain a normal shape, but in the womb, because of certain mechanical forces or restriction, there might be specific alteration in the morphology of the organs. For example, club feet, which is formed due to compression in the amniotic cavity. If the amniotic cavity is not big enough for the embryo to grow, the feet might grow in shape like a club. The feet might not be normal shape. So this is one of the example of deformation. In this particular panel, we can see how a club foot develops because there is not proper growth, proper space for the feet to grow. It becomes club shaped. Now, another type is syndrome. Now, what exactly happens in syndrome? In syndrome, a group of anomalies are occurring together, and the reason for the occurrence of this particular type of malformation is known, and the risk of recurrence is known. That means if a particular couple is giving birth to a child which is having this syndrome, by using the genetic factor, by using the law of probability, the risk of recurrence of this particular syndrome in the future progenies can be found out, and genetic counseling can be done of such individuals. For example, Down syndrome, fetal alcoholic syndrome. These syndrome, Down syndrome is caused because of because of the aberrant number of the chromosomes, chromosome number 21 in the individual. And fetal alcoholic syndrome is caused if the pregnant woman is having alcohol during the time of pregnancy. So these the cause of this particular malformation is known in the case of syndromes. For example, we can see in this panel fetal alcoholic syndrome, wherein a pregnant woman is taking alcohol throughout the period of pregnancy. She will give birth to a child which will be having certain specific features which can be recognized by their specific presence, specific pattern. As can be seen in this particular panel, these individuals will be having small head, they will be having epicanthal folds and underdeveloped jaws. So it can be seen that this particular malformation occurs because the woman is taking alcohol. Now another type is association. What exactly happens in association? It is a non-random appearance of two or more anomalies that occur together more frequently than by chance alone. That means more than one anomalies are occurring together in a number of individuals, and it is much more frequent than can happen by chance alone. But the cause of occurrence of such anomalies is not generally known. In syndrome it is known, but in association malformation we do not know the cause of such occurrence, and there is no known diagnosis for such malformation. For example, it is given a term VACTAL, which is vertebral defects, anal atresia, cardiac defects, tracheoesophageal fistula and renal anomalies. So this is a general term given for such anomalies and limb abnormality. Now recognition of one component of anomaly leads to search of other anomalies. So this is an individual which is having this particular association abnormality, and it is this particular individual is suffering from a number of anomalies in the entire body. Now these congenital malformation can also be caused by exogenous agents, and these are called as disruptions. And the agents which cause disruptions are known as teratogens. Sometimes a condition might be a disruption or malformation, and it is very difficult to find out what exactly is the cause of any anomaly. For example, Achondroplasia is a congenital defect of cartilage, and it is characterized by abnormal bone mineralization, which leads to underdevelopment of nasal cartilage and leads to shortened fingers. But this condition can also be caused by a defective gene on X chromosome, and it can also be caused by exposure of fetus to an anticoagulant known as warfarin. So it is difficult to find out what exactly is the cause for this particular condition in individual. Now, to what extent does the malformation occurs depends on the timing of exposure of the fetus to the teratogen, and these teratogens produce their effect during specific periods of development. Human development is divided into two periods: the embryonic period, which is up to the end of week 8 of pregnancy, and fetal period, which is the remaining period in which the fetus spend in the uterus. Now most of the organ systems are formed during the embryonic stage, and in fetal stage only growth and modeling of the organs occur. Now there is a sensitive period which is a broad period of time during the entire development, which is a sensitive period when the embryo will get exposed to teratogen. It will lead to formation of abnormal organ system. For example, if the if the embryo is exposed to alcohol during last trimester, it might lead to development of neurobehavioral effects or disorders. Whereas there is a critical period, which is a specific period of time during which if the embryo is exposed to teratogen, it will lead to a particular consequence. For example, malformation of ears due to exposure to thalidomide, which can occur if this embryo is exposed to thalidomide from days 34 to 38 of prenatal development. Before that and after that, this particular effect does not occur. So there are sensitive periods and critical periods. The period of maximum susceptibility is between week 3 to week 8 for human embryos. Prior to that, exposure to teratogen does not produce a significant abnormality, and if there is exposure, the embryo might get killed altogether. However, nervous system remains susceptible to teratogen throughout the development period, that is the entire period spent in the uterus. So this panel shows the period of time to which the embryo is most susceptible to teratogens. Prenatal death occurs from week 1 to 2. Week 3 to 8 is the most susceptible period for the embryo for teratogen, and after that there might be minor structural abnormalities and physiological abnormalities in the embryo. Now, certain factors which determine the capacity of an agent to produce birth defects have been described, and these are termed as principles of teratology. Susceptibility to teratogens depends on genotypes of the conceptor, that is the embryo and the maternal genome, the development stage or time of the exposure, the dose and duration of teratogen exposure, and the specific mechanism in which teratogen acts on the developing cells. And manifestation of abnormal development are malformation, disorder, growth disorder, functional disorder, and in these severe cases it leads to death. Now, teratogenic agents could be infectious agents like virus and bacteria, physical agents like radiation, drugs and chemical agents, hormones, and maternal metabolic imbalances, nutritional deficiencies, and male-mediated teratogenesis. Now, these teratogens, either they interfere with the nucleic acid, that is during replication or transcription, or they inhibit certain enzymes which lead to stopping of a specific pathway of development, or they might lead to decrease in the energy supply for the embryo to grow and develop. So these are some of the mechanisms by which these teratogens act on the embryo and produce malformation in the organs. Now, talking about infectious agents, there are a number of infectious agents which lead to malformation, and these are viruses and bacteria like rubella, herpes simplex virus, cytomegalovirus, HIV, and bacteria Toxoplasma gondii, congenital syphilis, which is again a bacterial disease. So these particular viruses and bacteria, they produce specific effects during specific period of exposure. Like rubella produces the most serious effect like heart defect during the first trimester, and it can also produce cataract, deafness, etc., and the risk of having impairment decrease with the time. If the embryo is exposed in the 3rd month, the risk of having impairments is only 50% as compared to 60 to 85% if the exposure in the first 8 weeks. Now, another virus like herpes simplex virus, it can cause severe impairments. About 23% of all adults have this particular virus, but only 1/10 are aware that they carry this virus, and a number of cases are reported each year. Now, 1 out of 15,000 births of individuals affected with this virus, the infant will also be having this herpes simplex virus infection, and the effects are that 1/3 of the infants will die, while 1/3 will be having permanent and severe brain damage, and others will be having mild impairment. And cytomegalovirus infection is the most common viral infection of the fetus, and during early trimester exposure, it leads to termination of the pregnancy, while later on it can lead to blindness, microcephaly, mental retardation, etc. Whereas HIV can be latent for many years in the individuals, and it can be transmitted from mother to fetus either prenatally at birth or through mother's milk postnatally. 30% of the pregnant women with HIV babies, the infants become infected with this particular virus and can lead to serious immunological deficiency. But if the woman is using the vaccine AZT, if the woman is using AZT treatment, then only 8% of the infant will be having this particular virus infection. Now, toxoplasmosis is basically, it occurs when there is infection by the Toxoplasma gondii, and if exposure in the first trimester, then severe central nervous system damage occurs. There is damage to the visual system, but if the exposure in the later time, then there will be increased occurrence of miscarriages and jaundices, pneumonia, etc. And it's in infection of syphilis, if the exposure during first and second trimester, then no effect occurs. If treatment is there, then there won't be any effect on the embryo. But in second and third trimester, it will lead to impairment of the organs of the embryo. Now, certain drugs and chemical agents can also cause these effects, like thalidomide, tetracycline, retinoic acid, diethylstilbestrol, and anti-epileptic and alcohol. Thalidomide can cause severe effect on the organisms, leading to malformation of ears, arms, etc. Whereas tetracycline can lead to yellow staining of the deciduous teeth, and it can also diminish the growth of the long bones. Retinoic acid, which is used for treatment of acne in women, it is vitamin A basically. Retinoic acid or vitamin A derivatives can lead to malformation like craniofacial dysmorphism, cleft palate, neural tube defects, etc. So, this is a panel which shows effect of retinoic acid on the embryo. Diethylstilbestrol, which is used to prevent miscarriages and premature deliveries, it can also have severe effect. If the exposure is prenatal, then it can lead to cervical cancer in the daughters which are born to this particular woman who is taking this drug, and it can also increase the risk of feminization in the males which is born to this particular woman. And there are higher rates of reproductive problems in both the sons and daughters of women taking diethylstilbestrol. Anti-epileptics also cause various malformations like facial clefts, nail dysplasia, microcephaly, etc. And taking of alcohol also leads to severe mental retardation, malformation, etc. in the progeny of the woman who is taking this particular drink. Ionizing radiation also leads to chromosome aberration, causing severe malformation in the individual. Now, maternal conditions like diabetes, etc., or congenital heart disease also have effect on the conceptors. And mechanical forces also lead to defects like dislocation of hip and club foot. Environmental teratogens like mercury and lead poisoning have severe effect on the embryo during development and will lead to malformation of the organ system. Now, male can also lead to teratogenesis if the male is exposed to chemicals and radiation, and the sperm which fertilizes the oocyte is having chromosomal defect, then it will also lead to problems in the embryo. Now, most common congenital malformation have multiple factors, and development of the malformation is dependent on a sum total of all the effect of these factors. So, this is all about how teratogens cause many malformation in the embryo during development, and different agents, different teratogens like infectious agents or chemicals or drugs, they have different way of acting on the embryo during development, and they produce specific results, specific malformation of organs and tissues which cause an individual, which lead to birth of an individual who is malformed and variously termed as monster, but actually is not so. It is just the effect of various teratogenic agents during the time of embryonic development which leads to formation of such individual. Thank you. I'll be talking about genetic errors of human development. Now, human development is a very finely tuned and finely balanced process which requires interplay of a number of genes at a specific time which get expressed at a specific time and which interplay with the product of other genes to produce a desired effect, a desired type of cell, desired type of tissue, organ system, organ and the complete individual. So, it is basically a very finely tuned process, and it also is dependent on the effect of the environment. It needs a specific environment for the embryo to develop properly. Now, what happens if the control system, if the message which is directing everything in the development of the embryo, if it goes awry, that means if there is some change in that particular message, what will be the effect on the development of the embryo? So, today I will be discussing the genetic errors of human development. What happens if the genetic message which is passed on from the parents to the embryo, if it is having certain defect or if it is changed, then what effect does it have on the development of the embryo? So, I will be discussing that in detail today. So, outline for this particular lecture, I will be talking about disorders which are because of genes, single gene, that is when only one gene is responsible for a particular malformation, single gene disorders, and when polygenes are responsible for a particular disorder, that is a number of genes are responsible for creating an anomaly in the individual. And then I will be discussing about autosomal disorders and sex chromosome disorders, how presence of extra autosomes or absence of a given number of autosomes can lead to specific disorders, and how presence or absence of specific sex chromosome can lead to formation of individual who is not normal. Also, I will be talking about epigenetic influence on the genetic control of development. So, the objective for this particular lecture would be, after this we should know what are the different genetic aspects related to formation of various congenital abnormalities, and I have referred a book known as Developmental Biology by Scott F. Gilbert for this particular topic. So, what exactly are congenital abnormalities? Congenital abnormalities are those abnormalities which are present in the individual since the time of birth. These abnormalities must have been present during the development of the embryo in the uterus of the mother. So, any abnormality present from the time of birth is termed as congenital abnormality, and this particular abnormality affects either a body part or a physiological function, and it might get remedied if it is a minor abnormality, or it might continue throughout the life of the organism of the individual who is born. Now, why this particular abnormality occurs? It occurs because of abnormal ontogenetic or abnormal embryonic development of the fetus. So, there is something abnormal going on which leads to formation of an individual with an abnormality. If everything is fine, if everything is under control, then individual will be born with normal organ system, normal physiology. But if there is something amiss, then that particular individual will be having certain malfunctioning of organ system. Now, this particular process of embryonic development is affected by genetic as well as environmental factors, and this particular process results from an interplay of these factors, that is environmental factors. And environmental factor does not mean the environmental factor surrounding the mother's body. Environmental factor basically means the environment surrounding the embryo or the fetus in the uterus of the mother. So, what exactly milieu is created in the amniotic sac of the embryo, that determines how the development will take place further on. Disturbance of the regulation and development cascades which take place during the development will lead to formation of abnormality. So, as I have already mentioned, it is a finely tuned process which requires interplay of number of genes. If there is any single disturbance, then there will be disturbance in the development cascade or the developmental steps which lead to error in the formation of cell and tissue. So, this is the etiology of the congenital abnormalities. The abnormalities which are present at birth, how they are formed. About the etiology or the origin of the 50 to 60% of the abnormality is not known. 20 to 25% abnormalities are because of multifactorial heredity. That means a number of factors are taking part in creation of such abnormality, from the genetic factors to environmental factors, all are taking part. Then chromosomal aberration lead to formation of about 6 to 7% of the congenital abnormality, and gene mutations are caused by about 7 to 8% of the abnormalities are caused by gene mutation, and only the environmental factors they are causing about 7 to 10% of the total congenital abnormalities in the individuals which are born. So, this has been taken from Clinically Oriented Embryology book, 6th edition, and the figures are from 1998. So, this is basically a general distribution that which type of abnormality lead to formation of abnormal individuals and what percentage contribution is there. So, we can see that chromosomal aberration and gene mutation together amount for about 13 to 15% of the abnormal individuals born. Now, how the genetic mutation and chromosomal aberration, how they cause malformation. This is what I am going to discuss about. Now, genetic factors is one of the common cause of congenital malformation, aberrant expression of genetic factors, and they account for about 1/4 of the entire malformation, as I have already seen in the panel. Now, this malformation could result due to single genes, effect of mutation in single genes, or it could be resulting from interplay of number of genes, that is polygenic influence. It could result from chromosomal aberration in the structure of chromosome or the number of chromosome, or it could be resulting from epigenetic, that is influence of the surrounding genetic material apart from the genes. Now, talking about single gene disorders. Single gene disorders are inherited as monogenic trait. That means one particular gene is deciding a specific pathway, and that particular pathway leads to development of a specific organ. Now, if there is a malformation, if there is a mutation in that particular gene, then the pathway which it specifies will not be carried out properly. If that pathway is not carried out properly, then the organ which is going to form by that particular pathway will also not be formed properly. So, this is a very simple cause and effect process. Now, there are several genes present in human body whose the mutation in which can lead to malformation in the body of the humans. And the genes, these genes, what are these genes? We can find out in the online reference system, that is Mendelian Inheritance in Man. This is an online database of different genes and what effect they have in the body of the human. So, it can be seen that there are a number of genes which lead to congenital abnormality in humans. Now, if it is known that a particular individual harbors a mutant gene, then the prenatal diagnosis can be done and genetic counseling can be done of such individuals, so that the pregnancy is managed effectively and there will be less chances of having an abnormal individual born to such couples. Examples of single gene disorders are Marfan Syndrome, Ehlers-Danlos Syndrome, Osteogenesis Imperfecta, Achondroplasia, holoprosencephaly, and xeroderma pigmentosum. And there are number of other disorders which are caused by a single gene mutation. Now, these figures which are shown along with these particular syndrome, they basically refer to the excess number of these particular genes in the Mendelian Inheritance in Man, which is an online reference system. And if we go to this particular system and type this particular excess number, then we'll get to see the sequence of this particular gene and abnormality in this particular gene, what will it lead to. So, talking about few of such single gene disorders, how exactly these disorders are caused by mutation in such genes. Talking about Marfan Syndrome. Marfan Syndrome occurs because of mutation in a gene known as FBN1 gene. This particular FBN1 gene is present in chromosome number 15 in the long arm, and it is denoted by 15q21.1. This is the location of this particular gene in the chromosome number 15. Now, mutation in this particular gene causes a disorder known as Marfan Syndrome. FBN1, this particular gene codes for a protein known as fibrillin-1. Now, as the name suggests, fibrillin-1, it codes for fibers. It forms thread-like microfibrils which become part of fibers of connective tissue. So, this the product of this particular gene FBN1 leads to formation of fibrillin-1, which is required for formation of connective tissue. Now, imagine a situation where this particular gene is not functional and fibrils are not formed. If the fibrils are not formed, then the connective tissue will not be formed properly, and connective tissue like cartilage and bone, if it is not formed properly, there will be severe effects on the systems like skeletal system, muscle system of the organism, and that happens in this particular syndrome, in which occurs a mutated form of this particular gene. Now, individuals with Marfan syndrome, they suffer from multiple anomalies. They usually are very tall and slender, and they have elongated fingers and toes, a condition known as arachnodactyly. And they have an arm span which exceeds their body height. That means they have so long hands, long arms that the total length of the both arms combined exceeds their total height. This is one of the recognizing features of this particular of the individual suffering from this particular syndrome. And two other primary features are presence of ectopia lentis, which is a vision problem caused due to dislocated lens, and defect in the large blood vessel which distribute blood from the heart to the rest of the body, that is aorta, defect in aorta. Now, as can be seen in this panel, the person suffering from this syndrome has very long arms, and apart from having these structural abnormalities, this individual suffers from other abnormalities like dislocated lens and abnormal aorta, which lead to various other problems, and these individuals face severe physiological problems in their life. Another example of monogene is Osteogenesis imperfecta. Now, Osteogenesis imperfecta is caused because of mutation in COL1A1 and COL1A2, CRTAP and P3H1 genes. Now, mutation in COL1A1 and COL1A2 genes are responsible for more than 90% of all the cases, because these genes lead to formation of collagen, which is again very important protein required for connective tissue formation. Now, as can be seen in this panel, the individual has it appears disfigured, the spine appears disfigured because of the malformation of collagen. Now, the genes, this COL-A1 and COL-A2 genes provide instruction for making collagen, which is the most abundant protein found in bone, skin and other connective tissue, which provides structure and strength to the body. If the gene is mutated, then we can very well guess the effect it will have on the body of the embryo. Another example is Achondroplasia. Achondroplasia is basically a type of dwarfism wherein the individuals who are suffering from this particular syndrome, they are having a very less height when they are born, and they do not grow to be very tall. They are basically dwarf individuals. This occurs because of presence of mutation in FGFR3 gene. This particular gene provides instruction for formation of a protein which is involved in development and maintenance of bones and brain tissue. Now, what happens if there is a mutation in this particular gene, the cartilage in this particular individual will not be converted into bone, and it will be happening particularly in the long bones of the arms and legs. If the bones of legs and arms are not formed properly, then the individual will be of very short stature and the individual would be a dwarf. As can be seen in this particular panel, all the people who are shown in this particular picture, they suffer from this particular disease or syndrome known as Achondroplasia, which results from a single gene FGFR3 mutation. Of course, they have these particular features: short arms and legs, enlarged head, prominent forehead, etc. And they have typically short fingers, though they can lead a normal life, but the stature is very short. Now, what about polygenic abnormality? We talked about monogenic disorders which are caused because of single genes. What about polygenic abnormality? These abnormalities are caused because of a number of genes which are acting simultaneously, or they might be acting in tandem or sequentially, leading to formation of a specific malformation. Now, polygenic inheritance means that there are more than one gene which affect the selected trait. Whereas similarly, multifactorial inheritance means that there are not only genetic factors but also environmental factors which affect the selected trait. So, we have to distinguish between these two. Polygenic means a number of genes affecting a given trait, and multifactorial inheritance means a number of genes and apart from that environmental factors which cause the formation of a given trait. So, it is not always very easy to distinguish between the polygenic abnormality and multifactorial abnormality. And today it is accepted that majority of the congenital abnormalities or anomalies, they are multifactorial. That means apart from having influence of the genes, the environmental influence also plays a very important part in formation of these anomalies. Now, coming to chromosomal abnormalities, that is abnormalities in chromosome, how they lead to development of anomalies in the embryo. Now, most of the chromosomal abnormalities, they lead to abortion of the fetus, but there are certain abnormalities which will not cause abortion of fetus but lead to formation of an embryo which is anomalous or which is having a large number of malformations. Now, the chromosomal disorders are related either to the number of the chromosome or they might be related to the structure of the chromosome. Now, how is there an aberration in the number of chromosomes? It might occur due to failure of chromosome to separate properly during cell division in meiosis, which leads the daughter cells to either have an extra chromosome or other daughter cells to have no chromosome of a representative set. So, that is how abnormality in the number of chromosomes is created. Abnormal structure or number of autosomal chromosome is usually incompatible with life because all chromosomes carry a large number of essential genes, and if a chromosome is lost, then the embryo will not survive. If there is an aberration in the number of autosomal chromosome, it will be termed as abnormality of autosome, whereas if there is an aberration in the structure or the number of sex chromosome, which are X and Y chromosome, it will be termed as abnormality of sex chromosome. So, all types are encountered in the development of embryos, and many individuals are born, though they are having aberration in the number or structure of chromosome, they are born and they survive to a specific age, and they carry out the normal function of any individual or normal individuals. Now, chromosomal abnormalities cause about 1/4 of the entire malformation, and they might be because of numbers and structural abnormalities. Now, talking about numerical autosomal abnormality, that is the abnormality in the number of the autosomes. There are some examples I have taken like Down syndrome, Edwards syndrome and Patau syndrome. Now, Down syndrome is a very familiar example. It results because of addition of one 21 chromosome, that is trisomy 21. Edwards syndrome results because of trisomy 18 and Patau syndrome results because of trisomy of chromosome number 13. So, all these chromosomes are present in one extra copy apart from their diploid number. Coming to Down syndrome, it can occur both in male and female, and it is a congenital anomaly which is caused because of presence of an excess chromosome 21 or because of translocation of chromosome 21. Now, the characteristic feature of this particular syndrome is that individuals are having a typical face and can be recognized at an instant. They have a prominent epicanthic fold above the eye. Most of the individuals are mentally retarded and they show excited muscle, that is non-excited muscle, that is hypotonia. They have very large tongue which is known as macroglossia, and they have a very prominent crease which is known as Simeon crease on their hand, and they also have eye and anomalies of the heart. Another example is that of Edwards syndrome, which occurs whenever there is trisomy 18, and it can both occur in both males as well as females. And again, it is a congenital chromosomal abnormality which can occur because of presence of extra 18 chromosome or because of translocation of a part of 18 chromosome. Now, the individuals who are having this particular syndrome, they show hypertonicity, that is, they show an excited state in the muscle, and they have a prominent occiput region, and most of them are mentally retarded, and the external ear, that is pinna, is very low set as compared to the normal humans. The external ears are set low in the face, and their hands are clenched and the fingers overlap with each other. So, these are some of the features of individuals suffering from Edwards syndrome. As can be seen in these particular pictures, these are the pictures of individuals who are afflicted with this particular syndrome. Now, another syndrome is Patau syndrome, which occurs because of presence of extra 13 chromosome and can again both occur in female and male because it is an autosomal disorder. Again, it occurs because of translocated chromosome 13 or an extra chromosome 13 present in the embryo. Now, there are variable defects in the facial development of individuals who is suffering from Patau syndrome. There might be cleft lip, cleft palate, or both, and the slope is usually, the head is usually small in size and the forehead is sloping, and there are various abnormalities of the eye, and there are severe abnormalities of the central nervous system. Again, there could be polydactyly and malformation of the heart and skin abnormalities, as well as abnormalities of the genitalia. So, this is one of the picture of individuals suffering from Patau syndrome who is having cleft lip. Other abnormalities can also occur in individual suffering from this particular syndrome. Now, talking about sex chromosomal abnormalities, that is the numerical abnormality, that is change in the normal numbers of the sex chromosome in an individual. A female is having given 22 pairs of autosome and X and X, that is 2 X chromosome, whereas a normal male is having 22 pairs of autosome and 1 X and 1 Y chromosome. If this particular X or Y changes, the number of X or Y changes in male and female, it will lead to specific disorders like Klinefelter Syndrome, which is caused by presence of an extra Y in a female, XXY, Turner Syndrome, which is caused by absence of an X chromosome, 45X, and triple X syndrome, which is caused by presence of an extra X chromosome in a normal female, and XYY, which is again presence of an extra Y in a normal male. So, in Klinefelter syndrome, it is caused because of presence of an extra X chromosome in the males, and males with Klinefelter syndrome are not usually detected in the newborn period, but as they grow during puberty, they start showing specific features which can tell us that these individuals are having abnormalities. These individuals have small testes and they show breast enlargement, and they are very tall in stature. Though they are males, they are having small testes and they also develop breast. And other features include mild mental deficiency and long limbs. And there are if there is an extra X chromosome, then it will lead to severe mental retardation and late sexual development and skeletal abnormality. Another syndrome is Turner syndrome, which is caused by absence of an X in female, and this whenever this occurs, it leads to ovarian hypoplasia, that is ovaries do not develop properly, or there is dysgenesis of ovary, there is infertility. The individuals are small in height, and they have a small, they have loose nuchal skin, that is they have webbed neck, and they have cardiac anomalies also. This is the picture and diagram of the individual suffering from Turner syndrome. Now, there is another syndrome which is known as triple X syndrome, which is caused due to presence of an additional X chromosome in females. The females are clinically normal and they are fertile, but there is an increased risk of learning disability and reduction in performance IQ and menstrual problems and early menopause in such females. These are the panels show pictures of females suffering from triple X syndrome. Now, there are structural abnormalities also like Cry-du-Chat syndrome, Wolff-Hirschhorn syndrome, and Dye-George syndrome. Now, Cry-du-Chat syndrome is one of the earliest syndrome which has been detected, and it is caused because of deletion in the short arm of chromosome number 5. The individuals have high-pitched cry, they have widely spaced eyes, small chin, small head, round face, and they show mental retardation. This picture is of individuals suffering from Cry-du-Chat syndrome. Another is Wolff-Hirschhorn syndrome, which is caused by deletion of short arm of chromosome number 4, and this results in broad and flat nasal bridge and a high forehead, and intellectual disability, and individuals suffer from seizures also. This can be seen an individual suffering from Wolff-Rayet syndrome. And there is another syndrome known as Diethylstilbestrol syndrome, which is caused because of deletion on long arm of chromosome 22, which leads to long face, high and broad nasal bridge, small teeth and downturned mouth and malformed ears. This is a picture of individuals suffering from Diethylstilbestrol syndrome. So these are the individuals. These are some syndromes which occur because of abnormalities in the number of chromosomes. Now, there could be abnormalities because of genomic imprinting or epigenetics, and two such examples I have taken. One is Prader-Willi syndrome and another is Angelman syndrome. Now, what happens in epigenetics is basically a given set of instruction is carried along with a gene. That is, instruction is imprinted in the genome of the mother and father, and this will be carried on from one generation to the next generation, and this will lead to formation of specific type of syndrome in the young one who is born. Now, in Prader-Willi syndrome is caused because of deletion or inactivation of part of long arm of chromosome number 15, and it is because of paternal origin, and it can also be caused by uniparental disomy, that is if only maternal chromosome 15 is inherited. Now, there is mild to moderate mental retardation in such individuals. There is hyperphagia, that means they eat a lot, and obesity also occurs. They have small hand and feet, and the gonads are small in size, and there is decreased mean height in adulthood in such individuals. So this is an example of panel which shows individuals suffering from Prader-Willi syndrome. Now, another example is that of Angelman syndrome. Now, in Angelman syndrome, it is caused because of deletion in the same chromosome, that is 15, and the long arm, but it is of maternal origin, that is the imprint is maternal, and it can also be caused by uniparental disomy if only the father's or the paternal's chromosome 15 is inherited. It also causes mental retardation and EEG abnormalities and ataxia and jerky arm movements. So this is an individual who suffers from Angelman syndrome. Now, what exactly are the nature of different nature of human syndrome? These syndromes could be caused because of pleiotropic effects, that is a production of several effects by one gene or a pair of genes, as can be seen in Waardenburg syndrome type 2, where there are defects in iris and pigmentation abnormalities because of malfunctioning macrothalmia protein, which is used as a transcription factor by all these cells and tissue, and several tissues get affected by the abnormal gene function. Or it could be relational pleiotropy, which occurs when several tissues are affected even though they are not expressing the mutated gene, but failure of expression of one gene causes non-differentiation of another one tissue, which leads to malformation of second tissue, which leads to malformation of the third tissue. So that is relational pleiotropy. Or there could be genetic heterogeneity, that is mutation in different genes can produce the same phenotype if they are part of the same signal transduction pathway. For example, cyclopia, which is presence of only one eye on the forehead of the individual, it can be caused by mutation in sonic hedgehog gene or by mutation in gene controlling cholesterol synthesis, because it acts through a protein known as SHH. The phenotypic variability could be caused because same mutation can produce different phenotype in different individuals, and this happens because gene interact with other genes in complex network and pathway. Now, these disorders could be diagnosed prenatally through various processes like examination of fetal cells, examination of amniotic fluid, ultrasound examination, and fetal cell DNA examination. And genetic counseling can be done of the parents who are more susceptible to have such malformed individuals, so that less of such individuals are born. So that will be all for the today's lecture. Thank you.
On that note, I would like to thank sir for this very enriching discussion, and thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.